Hello everyone, welcome to lecture 5 of our short course. In today's video we will talk about polygenic risk scores. We will discuss what a polygenic risk score is, how it is measured, and conclude with an essential issue in polygenic risk score calculations, population bias, which is essentially the heterogeneity problem I mentioned in the meta-analysis video from the previous lecture. So let's get started. Polygenic risk score, or PRS in short, is a measure of aggregated effects of genetic variants on the risk of developing a disease. So what's that mean? Let's decipher this sentence one by one. Now, when we talk about aggregated effects of genetic variants, we mean the genetic effects that are derived from GWAS studies. So we have effect size estimates for each genetic variant, and we aggregate them into a single score. That is the polygenic risk score. And that is a measure of individuals' risk of developing a disease. And this measure indicates a relative genetic risk compared to a reference population. And what do I mean with that? We have a population that we indicate the results are derived from the GWAS results. And therefore, when we calculate the PRS scores, those results are those results are indicating a reference population. And what happens in general in polygenic risk score calculations is that we calculate the risk score for all individuals, which would give us a distribution of scores such as this one. And then we define low-risk individuals and high-risk individuals. And when we say high-risk individuals, this is a high-risk individuals relative to a reference population, which is an important remark because this is different than defining an absolute risk. Absolute risk shows the likelihood of a disease occurring. Women, for example, women who carry a BRCA1 mutation have a 60 to 80% absolute risk of breast cancer. And this would be true even without any comparison to any groups of people. Whereas when we talk about polygenic risk score, because they are derived from a reference population, these scores are indicating relative risks. So these are different than absolute risks, and they have to be interpreted by health professionals. And although that is different than absolute risks, these are still relevant for clinical applications, and they have so much use in clinical settings. I will not talk about the applications of polygenic risk scores, but rather will be focusing on how they are calculated and the population bias issue in them. But that has to be noted that it is not an absolute risk, and therefore when we say the high-risk individuals, we mean that this is compared to a reference population. It doesn't necessarily mean that you have a high percentage of absolute risk of getting a disease. So it is completely different. Now, that being said, now in the next slide, I will explain the core logic of how we are calculating polygenic risk scores. So this is a figure taken from a recent study. It explains the core logic of the calculating polygenic risk scores. Excuse me. So what we does is that we have here polygenic risk score is calculated for each individual, and there are two components that matters here. One is the effect size estimates that are derived from GWAS studies, and the other one is the genotype values or genotype dosage for individuals. And we take these values for different parts of the genome and we aggregate them into a single score for each individual. And one important remark when we are calculating polygenic risk score is that we take the effect size estimates from something called a discovery cohort. Whereas when we calculate the polygenic risk scores, we calculate them for something like called target cohort, which is independent from discovery cohort. So we have GWAS results from one cohort, and then we calculate the polygenic risk scores on different cohort. And these two cohorts has to be taken from the same population. And I will explain why this is important in the next couple of slides when we talk about population bias. But the idea is that when you have the GWAS results from the discovery cohort, you have different SNPs, and you know the risk alleles and their effects, which are the weights. And then you have on the target data individuals, their genotype informations, and you just consider their dosage, genotype dosages for each individual, for each location, and then multiply it with the effect size estimates derived from discovery cohort. And then this indicates for each individual a single score, polygenic risk score. And this is how we calculate the polygenic risk score. But there is one important issue here, which is the LD issue between genetic variants. So if you have a genetic variant which is in an LD block of, let's say, 100 other SNPs, then when you calculate the polygenic risk score, you are adding the same information or similar information 100 times over and over again, because those SNPs are in high LD. Their effects are similar, and therefore you are adding similar effects over and over again. And this indicates a bias for markers that are in large LD blocks compared to the variants that do not have much LD structure with other variants. Therefore, when we talk about polygenic risk score calculations, what we do is simply something called C plus T approach, which stands for clumping and then plus threshold. And I will not go over the details of this, but there is a very good tutorial that you can go in your own pace to learn how to do polygenic risk score calculation in Plink or other kind of software. And the idea is that you divide the genome into windows of SNPs, and then for each windows you apply a threshold clumping. The idea of clumping, which is you apply a threshold of, let's say, for example, 0.1, and anything, any pair of SNPs that any SNPs that have R square less than 0.1 with the index SNP is removed, and this would filter the SNPs with high LD. So therefore you don't add the similar effects due to the high LD over and over again in the genotype in the polygenic risk score calculations. And the second part is the thresholding. So rather than adding all genotype effects in your polygenic risk score calculations, you only consider the genotypes that are highly associated with the trait of interest. And maybe I should have said, when we talk about the polygenic risk score, we are talking about risk score for a given trait, right? And therefore, when we apply a thresholding step, we take the SNPs that are highly associated in a GWAS study with a trait of interest. And rather than analyzing the all aggregated results of all SNPs, we are only considering the SNPs that matters for the disease. So this is the most common approach. On the right, we have a table of available methods for calculating polygenic score. There are so many. This is not the exclusive list of methods. But the core logic is basically, you can think about it like a C plus T approach. So I will not talk about other methods because the idea is just giving you the core logic of how PRS are calculated. And in the next couple of slides, I will be talking about this population bias issue. So this figure is taken from a fairly recent study by Martin, Kanai, and Kamantani et al. Clinical use of current polygenic risk scores may exacerbate health disparities. And here they show that in the figure that the polygenic risk scores are predominantly calculated by European individuals. So this indicates a high population bias, because if you don't have in discovery cohort a European GWAS, if you don't have GWAS studies for, let's say, East Asians, then you cannot really apply PRS from polygenic risk scores for East Asians when you don't have the discovery cohort for East Asians. You have to take polygenic risk scores from, let's say, European individuals, but that would create a population bias that I will explain in the next slide. So because the results are mostly European, it defines, it sometimes creates results, polygenic risk scores that are taken from different populations. And why this causes a problem? Here there are so many figures that I will go one by one. So again, taken from the same study, these figures, if you look into the top left, there are three different populations that they have considered in their study: African populations, European, and East Asian populations. And on the right, we see the different minor allele frequencies between populations. And on the bottom, we have C, D, and E plots where they looked into LD pairs between populations. So they look into genotype, one genotype location from East Asian, and then the other genotype from African population, and they look at the LD structure between them, and they do this for different pairs of populations, and you can see that different pairs of populations have different LD structures, and this would strongly indicate that the polygenic risk scores will be affected because the LD structure is simply different. And on the right, they show their experiment on how this population bias is causing health disparities in polygenic risk score estimates. So if we focus on this A plot, we see that they looked on two different discovery cohorts. They consider European individuals and individuals from Biobank Japan, East Asian individuals. So there are two different discovery cohorts they considered. And then they considered three target cohorts: European individuals, East Asian, and African individuals. And they looked into multiple different cases for calculating polygenic risk score. So for example, if target cohort is, if we focus on this target cohort, European individuals, they calculated the polygenic risk scores of European individuals in two different ways. One is they calculated their polygenic risk scores by taking GWAS results from European individuals as their discovery cohort, and they also calculated polygenic risk score by taking East Asian individuals' GWAS results as their discovery cohort. So this defines two different two different polygenic risk scores for European individuals. And here in these five graphs, they show you the differences between the polygenic risk scores. So for example, if we focus on this plot D, these are the individuals' polygenic risk scores for European UK Biobank individuals. And here we have red and blue, where red is polygenic risk scores calculated by European individuals, where the discovery cohort is also European individuals. So they are taken from the same cohort, same population, sorry. And the blue bars are indicating polygenic risk scores calculated for European individuals, but the discovery cohort is from East Asian Biobank Japan individuals. And you can see that, for example, if you focus on this height, the polygenic risk scores that are derived by using the European GWAS summary results explain roughly more than 10% of the trait. Whereas if you focus on the polygenic risk scores calculated for European individuals using the GWAS results from East Asian individuals, they only explain roughly 3% or something. So there is a huge difference, and it is persistent across different traits of interest. So this indicates that when you look into polygenic risk scores calculations, you have to consider that the discovery cohort and the target cohort are from the same population, because if you change the population, then because the LD structure changes, the polygenic risk scores are affected, and it becomes worse, as you can see on this plot. And similar results are shown in other plots as well. So this is also showing you the heterogeneity issue between populations. Remember that in the previous video where we talk about the meta-analysis issues, there was a meta-analysis study about height which combined 5 different ancestry cohorts, and which would they used the fixed effect models. They did not use the random effects model, but here you can see clearly that there is a high heterogeneity between the populations that cause significant difference even in the polygenic risk score estimate. So therefore, we should not ignore the population differences, and we should consider the heterogeneity carefully between populations, and this also affects the population, the polygenic risk score estimates. Okay, so we talked about the population bias issue, and this brings us the end of today's video. We briefly covered the polygenic risk scores and how they are calculated, and the population bias issue in their calculation process. In the next video, I will be talking about heritability. What is heritability, how it is being measured, and the challenges in heritability studies. So thank you for watching, and I hope you enjoyed the lecture. I will see you in the next video. Bye.